Okay, welcome everyone, and welcome to the very first session of the 2022 Biocontrol Technical Workshop Series under the ASEAN Fall Armyworm Action Plan. And we're very happy to be hosting this series in partnership with CABI. My name is Alison Watson and I'll be moderating the session. And we have a fantastic presentation lined up. I'm really excited with what's going to be presented, and we have some fantastic speakers joining us as well that you can see on screen, or people helping out with questions that you may have in the Q&A. So before we start, I'd just like to go over how we interact in today's session. It's really important to use this opportunity to ask all those burning questions you have around BT biopesticides, because you have the experts here. So please put all your questions, the Q&A, in the Q&A box, and we'll try and move through those. It's much easier if you use that rather than the chat, because it means I can see all the questions and make sure that they are asked. But please use the chat box to introduce yourself. If you have any resources that you would like to share, also you can post them in there, and you can also make comments in there as well. Just be polite, and if you'd like to congratulate one of the speakers or say something, please do so as well. Now, just a reminder that this is the first session of the series. We had a very big series last year, so we thought we would this time do another series, but really focus in on that specific technologies and approaches, and really look at practical information and resources in the field. So our session today on BT biopesticides, for example, has a lot of practical information. It's also going to be followed up by a second session on June 30 that will take us into the field to get a closer look of how to use BT in crops with field examples. And you'll be able to register if you haven't already at our events page. Now, just a reminder as well, if you've got any further questions after this, or you want to share some work that you're doing, in particular in relation to BT biopesticides, you can participate in our forum, and you just really have to go to our ASEANFAWaction.org website, go to community, click on forum. You'll see a little forum there under Biocontrol, and you can actually ask your questions in there, and we'll actually get some of our experts to answer those. So don't worry if your question doesn't get answered today. There is room for it there as well. And we do issue certificates of participation, but you do have to participate in our forum discussion if you want a participation certificate. Now, just a quick introduction. We're really lucky to have three speakers today join us through the session. We've got Dr. Roma Gwyn, an independent international expert on biocontrol and lead of our upcoming Biocontrol Masterclass Research and Regulatory Series. Dr. Daniel Zomick, Technical Development Specialist by the Biorational Crop Protection Division at Valent Biosciences, and Fang Zhang, Regional Director, the East and Southeast Asia of CABI, and he'll be joining us at the end to give us a bit of a summary of the session. Now, before we start, we want to find out a little bit about who's in the room. So we've got 4 questions, and we wanted to find out a little bit about your knowledge. So I'm just going to launch the first poll. Hopefully you can see that. I'm afraid I don't think our panelists can participate in the poll. So sorry there if you've got a burning desire. But look, you've got 4 questions. You should be able to see that. Can people see that coming up? Yep, I've got some people answering. So the first one is, what stakeholder group best describes the organisation you work for? So you've got civil society, government, international organisations, research, academia, and other, for those people who are also very keen on biocontrol but don't fall into those categories. And look, here's a really interesting one. We wanted to know who in the room has used any biopesticides before. Okay, we're getting a real mix there. And have you used BT before? Another kind of interesting results. I'm getting a preview here, so I'm sneaking into the answers already. But the 4th one is, have you actually treated for fall armyworm with any technique or approach? We really just want to know that, have you actually been in there and been involved in trying to control fall armyworm? So that's getting a mixture of results too. I'm going to give everyone maybe 10 more seconds. You've got a few questions to answer there. We've got 70 replies so far, so that's pretty good. So keep them coming, everyone. And we've also got more joining the room. So we're up to 138. So that's really good. It's great to see everyone. And I'm going to end the poll very shortly. And I'm going to share the results. So hopefully you can all see that. Daniel, you've got a bit of a nod to, can you see the results? So what stakeholder group? We've got 45% from private sector there, but strong government as well, and then a mix of others. Nice to see the research organisations. Have you used any biopesticide? Never, 33%, occasionally 31%, and often 17%. So we've got a bit of a mix actually there as well. So that's good to see. Have you used BT? Oh, here we go. We've got quite a nice spread. Never is 40%. So we've got lots in the room that are really wanting to know what is the deal with BT and how can we use it and what does it do? And then we've got 20% there that often. So I think you're going to have some questions, Daniel, from both ends of the spectrum. And have you treated for fall armyworm? This is interesting too. Wow. Okay, 54% no, but 46% yes. So that's fantastic. So that's going to be a really good discussion. So look, there's lots of questions I can see already coming through probably. So please ask those in the Q&A as we go through. Now, thanks for doing that, everyone. That sort of sets us up for a good discussion, and it also sets us up for introducing our speaker, Daniel Zomit from Valent Biosciences, where he is responsible for commercial support and product training on the BT portfolio. Valent Biosciences is one of the largest BT manufacturers in the world, with products registered in more than 65 countries. He's going to be joined by Dr. Christopher Moses, and I also see Katie there as well from Valent Biosciences. Christopher is actually the biorational expert for Sumitomo Chemical, which is the parent company to Valent Biosciences in Asia. So they will be helping to answer any technical questions that come through. So please keep them coming through because they are there and ready to answer your questions. So without further ado, I'd really like to introduce Daniel, bring a wealth of experience. It's great to have you today, and we're really interested to have this presentation on the management of fall armyworm with sprayable BT. So welcome, Daniel. I'm just going to turn my video off and I'm going to ask you if you could load your presentation.
All right, thank you so much. I think you have to stop sharing your screen.
Yep. Perfect.
Here we go. All right, you can see it?
Yep, just need to maximize it.
All right, there we go. Well, excellent. You know, I want to start by thanking Allison and the Grow Asia organization just for this great opportunity to introduce you all, or maybe remind you about some interesting things about Bacillus thuringiensis. Today, more than ever, commercial BTs are a really important tool, not only for managing fall armyworm, but also for other caterpillar pests. As society moves towards a desire for using safer, lower risk pesticides, having products like BTs on the market is really critically important for our future. And so I'd like to start maybe stepping back a little bit with just a reminder about what a biocontrol is. We are very much in a very distinct group here in biocontrol, in which biopesticides are defined as maybe macroorganisms like beneficial insects, microbes like what we'll talk about today, natural substances like plant extracts, and semiochemicals like pheromones. And actually, Roma gave an excellent introduction to biocontrols. If you go to this link here, the Fall Armyworm Action website in the video section, there's a really good introduction to all of these different biocontrol tools. But today we're going to focus really just on the microorganisms. And in general, there are three general classes of microbial bioprotectants. The first are fungi. These are microbes like Metarhizium or Bavaria bassiana, bacterium like BTs, or viruses. Now, this image here on the left shows the different ways in which these different classes operate. So, for example, BTs and viruses exclusively work through ingestion, while fungi can bind to the fungi will attach to the outside of the insect and actually invade them from the outside in. And that's really an important aspect about microbial biocontrols. It's that they operate very differently from chemical controls. So for example, microbial biocontrols are microorganisms that may or may not have secondary compounds, while chemical controls are very defined active ingredients. Microbial biocontrols are often have a complex mode of action. So for example, BTs, we'll talk about it as a stomach poison today, while chemical controls have a very singular mode of action. For example, having a very discrete target site. Microbial biocontrols can be really difficult to define what the active substance is, as it's the concoction of lots of different things. While chemical controls always have a very clearly defined active ingredient, active substance. Microbial biocontrols also oftentimes are slow to have its effect, slow to kill the insect, while chemical controls can be very quick, having very good knockdown activity. And so what I think it's important to mention here, and what I want to draw you to, is the fact that when we're talking about microbial biocontrols and about BTs specifically, I want you to think about them not in the same vein as you would traditional chemical controls, but to consider that it is a living organism and it operates within the insect in a different way. And why it's important to keep that in mind is because that also then helps inform us about how to use the products most effectively, and to use that natural mode of action to our benefit. So Bacillus thuringiensis is very much a targeted bacterial disease of caterpillars. If you look on the left-hand side here, this is a single cell of BT, which contains a viable spore that's kind of like synonymous with a seed from a plant. And there's that beautiful crystal shape, that bipyramidal shape, which are the cryproteins. So these are crystal, they're named after the shape, the crystal proteins, which are actually insecticidal proteins that will naturally crystallize within the cell. They're actually produced at such high quantity and at such purity during the fermentation process that it'll just naturally produce these crystals. The primary mode of action is to form pores, which will then inhibit feeding and eventually kill the insect. BTs also contain a viable spore, which, as I mentioned, is synonymous with a seed from a plant. These spores will germinate in the insect gut, not on the plant or in the bag or the bottle. It does contribute to mortality, but as I'll show you later today, the spores themselves are actually not toxic to the caterpillars. There are other soluble synergists, some of them directly toxic, some of them not, to caterpillars. For example, VIP3A, the vegetative insecticidal proteins, has a similar mode of action to cryproteins, but a very unique target site. It's also soluble and doesn't form those beautiful crystals. There's also secondary compounds that can synergize cryproteins, like PD100, another name for zwittermycin, and other enzymes and byproducts that are produced by the bacteria. So here is just a perfect example of how complex these how complex this product can be. So when we consider what actually goes into the sprayable BT, so what goes into the formulated product, it's all of these different components put together. And just to sort of to drive that home, this is an SEM image of what the components of the commercial BT look like. And if you'll see, there's sort of a rounded, there's these rounded objects, those are the spores. And if you look closely, you can find those beautiful bipyramidal crystals just spotted throughout. And so in a commercial BT product, we have a very concentrated, a high concentration of these spores and crystals, which we're spraying out onto the plant. Obviously, you can't see the soluble components here, but this is really the bulk of the products when you look under a microscope. Now, BTs are really commonly used throughout Asia, throughout the world. And they may, in fact, be more common than you might think for those folks that haven't used them before. For example, BTs are commonly used in cabbage or leafy greens as an important resistance management. So that is, as insects become intolerant to other insecticides, commercial BTs can still effectively control them. In the case of pineapples, for example, commercial BTs are used as they do not contribute to chemical residues on the fruit, ensuring that there are no issues with exporting those produce. In the case of oil palms, there's a really interesting and unique use of BTs to control the bunch moth, caterpillar that will eat the actual fruit themselves. And in this case, there's an important pollinating weevil. The targeted activity of these commercial BTs can actually very efficiently control those caterpillar pests, but have no effect on the pollinator weevil because they have such a selective mode of action. And finally, mosquito control. The commercial BTI, the Israeliensis strains are actually are incredibly are safe enough to be used actually in populated areas, in drinking water and things like that to control mosquitoes, the mosquito larvae that can survive in that standing water. So this way, commercial BTs may be more common than you might have thought. And actually, they've been around for a very long time. So commercial products have been used for more than 50 years. Last year, we celebrated, and today we're still celebrating, the 50th anniversary of Dipel. That's the flagship BT Kerstaki strain sold by Valent Biosciences. It was first registered back in the 1970s. And so today we're celebrating 50 years. And if you think back at what chemistries were around 50 years ago, few, if any of them, have really survived and are in common usage today. And that's because there's been a shift in what we want out of our pesticides. And that shift has really maintained commercial BTs like Dipel into today. And also today we're really distributing more BTs than ever in our history, which just goes to show that that the demand for these lower risk targeted insecticides is really high, and that farmers are seeing the efficacy. Now, with respect to fall armyworm, commercial BTs have been used as a tool against fall armyworm in the Americas really for decades. Excuse me, the fall armyworm is native to Central and South America. And so actually in the early applications of commercial BTs in the United States, early applications of Dipel, were oftentimes targeted to corn, where they could be used to control the European corn borer, but also other incidental pests like the fall armyworm. In Brazil, back in the 2013-2014 season, more than 4 million hectares of corn, cotton, and soybean were sprayed with commercial BTs to combat the invasion of the Helicoverpa armigera. And so, these products are really well known within the Americas to control caterpillar pest in corn, cotton, and soybeans. And so, I think by the end of this session today and the next one in June, I hope you have a pretty good idea about how they'll be able to be used effectively throughout Asia. Now, there are a number of common reasons why folks will use commercial BTs. The first is that they are effective. We see consistent efficacy, and if used at the appropriate time and at the appropriate rate, they can be similar to chemical controls. They have a low environmental impact as they're safe for beneficials, and also they are a natural part of the environment. BTs are really all around us. They have an excellent worker safety profile, which means it's low risk for applicators. And they're also important for chemical residue management. So there's no maximum residue levels anywhere in the world. And finally, and maybe most importantly, in the case of fall armyworm, they're an important tool for resistance management, as commercial BTs have shown no cross resistance with any other insecticide class. Period, full stop. It's really remarkable how these products are so different. So you may recognize some of these attributes as things that are important to you, but what it really comes down to is that we have this a very effective but natural substance that that represents a low risk for applicators and has a lot of implications for how produce can be sold and used. Now, Bacillus thuringiensis in agriculture actually gets very specific, so down to the strain level. So what you'll see when you look at a BT label is you'll see that all the products are labeled as Bacillus thuringiensis, but there are two subspecies you'll often see. These are either the Kerstaki or the Azawai type strains. Now, within these subspecies, there are individual strains and then production strains isolated from those. So when you buy a bag of Dipel or a bottle of other BT products, it is only a singular microorganism that's in that bottle. So we produce at 100,000 liter scale a single microorganism, kind of like you would ferment beer. You're growing that singular yeast. Now, each production strain has a unique mix of insecticidal components that I'll go into more detail with momentarily. And because those components can be so different between strains, each one is actually registered as an individual active substance. So the strain ABTS 351 is considered a different active substance from the 1857 strain. And that's because those attributes are so different. And that's another reason why we really need to understand the complexities of microorganisms and consider them differently than chemicals, in that each one has its own unique properties, its own unique value. Now, there are different subspecies used for other uses. So, for example, we've talked about the Kerstaki and Azawai type strains that are used for caterpillar control. While there's a subspecies Tenebrionis, the BTT, that's used for controlling beetle larvae. The BT Israeliensis strain is used for mosquito and blackfly control. However, this is very important. Only the subspecies Kerstaki and Azawai can be used to control the fall armyworm. Oftentimes, in my experience, there can be a misconception about which of these types of strains can be used for what types of insects. But if you just keep in mind that only Kerstaki and Azawai type strains are effective against fall armyworm, you may find the Israeliensis out in the market, but it is not at all for this particular use. Now, these commercial BTs are actually effective against almost all agronomically important pests. I have here listed out just some of the pests that are on the commercial labels for the Sumitomo chemical products that are available throughout the region. For example, diamondback moth, nettle caterpillar, or bunch moth on the oil palm, as well as cotton bollworm, beet armyworm, and of course, fall armyworm. Now, there are a few insects that are not controlled by commercial BTs, and those are pests that either immediately burrow into a plant or those that are actually hatched within a plant. And that's because commercial BTs are not systemic. You saw in that image at the beginning, these are just actual crystals and spores, a physical thing. Not necessarily a soluble metabolite. And so for that reason, they cannot move within the plant and they cannot move across the plant. So the insect will have to eat the foliage on the outside to actually ingest that commercial BT. And so after that brief introduction, I'd like to just open it up for any specific questions.
Great, thank you, Daniel. Excellent initial start there, and there's already lots of questions coming in. I can see Chris is pretty busy, so I may take a few questions that Chris has asked as well, just so that everyone has a bit of a discussion on them. I think the first one here was that you mentioned BT are used to manage pests with resistance to chemical insecticides. How about resistance to BT proteins? How is that managed?
Yeah, so that's actually a very interesting question. It turns out that individual cry proteins, as I'll kind of describe later, can in some cases bind to entirely different receptors. So for example, resistance to one cry protein may not necessarily confer resistance to another cry protein. And this is really important with respect to genetically modified crops, where those crops will often contain fewer cry proteins than what you would find in the commercial BTs. So for example, in Brazil, the Intacta soybean contains a high concentration of cry 1AC. which effectively controls most pests, but leaves the armyworms out in the field. And so in that case, using a product like an azawai type strain can actually effectively control those because, as I'll show in a minute, the Cry 1C and 1D are so unique from that Cry 1AC that they can actually control those insects better. So they can be complementary. But it really does depend on the BT crop. But they can be complementary technologies.
Excellent. Here's another question, a good question here. There are various strains of BT in the market. Will it be more efficient for a region to use their indigenous strain, or doesn't it have a huge difference between strains?
That's also a very good question. The commercial strains that are being mass produced were specifically selected through these screening programs. And I think what maybe Roma could step in on this one, but I think the really important aspect here is the regulation of these commercial products. While it may, in some cases, while there may be other better strains out there in the market, the regulatory rigor, the required, the study that has to go into getting a commercial strain onto the market is immense enough that we really have to pick and choose.
Yep.
Yeah, so Dipel and Zentari, for example, are registered in 65 different countries. So that means, yeah, it's an immense amount of work.
Good answer. And actually, I'll just throw that over to Roma, and then after that, we'll move on because there's lots of questions coming through. But I know that some of those questions will be answered further down, and we'll have time at the end and other questions times. Roma, any feedback on that question?
Sorry, could you just repeat the question?
The question was, there are various strains of BT in the market. Would it be more efficient for a region to use their indigenous strain, or doesn't it have a huge difference between strains?
No, I mean, Daniel's right that so the registration of a new microorganism is going to cost in the region of getting up to, with product development process and the registration itself, you're looking at a spend that's 5 million plus. So if you're developing a new strain, you have to think it's better than what's already in the market to justify that kind of expenditure.
Yeah.
And then from a biological point of view, there's nothing to indicate that the regional co-location of a strain with a pest, it makes it better. What you can have with some microorganisms is their adaptation to temperature. So for example, if you're working in a really dry country and a hot country, you might want a strain that's better in dry and hot conditions. Yep. But a company who's developing the products will know that, and they'll filter for that, and they'll choose the right strain for the right conditions where they're planning to sell it.
Okay, great answer. Thanks, Roma. Daniel, I'm going to let you go on because I know that we've got lots to get through, and you've got some great stuff coming up.
Excellent. So next, I'd like to talk about the mode of action of commercial BTs. And so we'll start with a video. So commercial BTs work exclusively through ingestion. The caterpillar larvae has to actually consume the crystals and spores. They can't absorb it through their skin. Now these crystals and spores, the crystals will then dissolve in the uniquely high pH of the the high pH of the caterpillar midgut. This releases those cry proteins, which are actually activated by the insect's own digestive system, which is kind of cool. BTs really use the insect against itself. Once activated, those proteins will then form these pores, which, when inserted into the membrane, cause the cells along the midgut to rupture. This allows BT spores and other microbes that are in the stomach to enter the hemocyle, or the insect's bloodstream, and will actually kill the insect through sepsis, through blood poisoning. So in fact, BTs really colonize the insect from the inside out. It's kind of grotesque, but really fascinating. And if you do find intoxicated insects out in the field, you'll find these brown, shriveled up insects because they've essentially been colonized by all of the microbes within the bug. Now, this mode of action is quick to stop feeding, but slow to kill. So this is a lettuce feeding assay. So these are treated lettuce leaves with either Zentari, that's a BTA product, or chlorantraniliprole, the Rynaxypyr technology. This is with second instar diamondback moth. And if you'll notice in the upper right-hand corner, there is the hours or days since application. So I'm going to start this over again. And what I'd like you to focus on is that even after 24 hours, in both treatments for the Zentari and the chlorantraniliprole treatments, insects are still moving around on the leaf surface. However, in contrast to the control, they're not feeding. And that's because when the cryproteins bind to the stomach and form those pores, it actually neutralizes that alkaline stomach and prevents them from being able to digest their food, to eat anything else. And so for that reason, that feeding cessation will occur very quickly, within minutes to hours after intoxication, while actual mortality, actually killing the insect, can take anywhere from a day or two, because that complete mode of action of colonizing the insect and working its way through their system just takes time, just like it takes us a few days to get really sick after we catch a cold. And that's important to keep in mind because in the case of chemical insecticides, oftentimes you can go out the next day and look to see if you have effectively controlled your target pest. But in the case of commercial BTs and really biocontrol solutions in general, when validating whether an application was effective or not, you really want to give the products sometimes time to work and time to control those insects. Now, BT strains have a very unique protein profiles. This is by far the most important difference between commercial strains. And that's because these cryproteins are the tip of the spear. This is the primary mode of action of the commercial BTs. Now, you'll notice I have here the two commercial strains sold by Sumitomo Chemical. The one on the left is the dipel strain, the subspecies Kerstaki ABTS 351. And on the right is the subspecies Azawai, ABTS 1857. And if you look, it's kind of color-coded, but on the left-hand side, dipel is characterized by a Cry1AA, 1AB, and 1AC. These are all relatively similar cryproteins. It also includes the Cry2AA. Now, this nomenclature, it looks a bit strange, but it actually denotes how similar these cryproteins are to each other. There are in fact hundreds, if not thousands, of different variants of cryproteins that have been found in nature. And to understand the relationship between these different cryproteins, we give them these indicator number and letters. So Cry1AA and 1AB are very similar to one another, while Cry1AA and Cry2AA are very different and actually bind to completely different receptor sites in the insect midgut. Similarly, in Zentari, while it does contain the same Cry1AA and 1AB, it uniquely expresses Cry1C and 1D. Now, these actual cryproteins, that actually defines really the Azawai type strains. That 1C and 1D also have unique binding sites, not only from one another, but also from the Cry1A type protein. So as I mentioned previously about genetically modified crops, insects that may not be controlled by a Cry1AC, for example, can be efficiently controlled by the Cry1C or Cry1D. And that's because different pest species actually have variable susceptibility to each protein. I mean, it's truly fascinating. This table on the left-hand side shows different pest species. And along the top, all of the different cryproteins that are in commercial BTs really around the world. Most of them only have these 6 toxins. Now, as you'll see, the pluses and minuses represent relative susceptibility. A minus doesn't mean that the insect is not susceptible. It just means that it requires a lot more of the material to kill the insect. And if we focus down on the one on the bottom, the fall armyworm, you'll see that while Cry1AB, which is the major component in both the BTK and BTA strains, most BT strains for that matter, can be somewhat effective against this species. Cry1C and 1D are actually acutely toxic to the fall armyworm. And this leads to a very fascinating example of where Bt subspecies matters quite a bit when trying to control an insect. In general, across other insects like the Helicoverpa or the cabbage looper, the Trichoplusa knee, generally an Azawi or Kursaki will control them essentially the same. There's not usually significant differences, but there are those rare instances like with Plutella and with Spodoptera species where the subspecies makes an important difference. And just to round it out, the reason for this differential susceptibility is that each insect has actually different receptors. And so that just goes to show that there's really the importance of the lack of cross resistance between some of these is based on that. Now, this is an example of this is a study from Laura Becerra and Associates, in which fall armyworm populations were collected from cornfields across Mexico. So this designation down here along the bottom is actually referencing different cornfields across Mexico. On the left-hand side, you'll see that this is the LC95. So the amount of product that was required to kill 95% of larvae in the study. And so these values can be quite high, but that's only because this is for complete mortality. And you'll notice across the board that while both Dipel and Centauri were very effective at killing the insects and controlling these insects, the BTA Centauri was more effective, and that's because of the Cry1C and Cry1D. Now, I wanted to show this also because it really shows how important it is to respect the application rate on the label. Commercial BTs, you'll notice, may have relatively high application rates compared to other chemistries. So you'll be using half a liter of a BT versus maybe only 100 mils of a chemical insecticide. And that's because even though the crystals and spores are very concentrated, you really need to get enough of them out into the field to efficiently cover the whole crop to effectively protect it. And so, especially in the case with commercial BTKs, these products can be very effective against fall armyworm. However, it is important to respect the application rates. While they may seem high, it's only because of this unique property where it does require slightly more of the BTKs to control those insects. So just to be clear, both of these products will work against fall armyworm, BTK and BTA. However, there is this slight difference that does give the benefit to commercial BTAs. And I really only know of maybe one or two other examples where this is actually the case. It's quite fascinating. Now, BTs have an entirely unique mode of action, which is really critical for resistance management. So unlike chemical insecticides that will have typically a single target site mode of action, complex mode of action of BTs means that you have a number of different components that are all working separately. So for example, the Cry1AC, Cry2As will hit different receptor sites, and the spores will also contribute, hitting with a different kind of effect. So commercial BTs are an IRAC group 11 that refers to stomach poisons. And actually, this is a cross section of a caterpillar. So if you cut the insect, across its body, and this is a caterpillar that was treated with a Cry1AC that has a red label to it. So anywhere that you see the red color is actually where that Cry protein bound to the inside of the midgut. So you can see in this case that it's very efficiently covering and will cause all of those cells to swell and rupture. In fact, you can see some of those cells are starting to swell already. Now, this target site is entirely unique from all other insecticides. The only other insecticide that has an effect on the midgut are viruses. And so for that reason, using commercial BTs either in a tank mix or rotation can be incredibly valuable in the long run to protect, the insecticides that you'll need to rely on even for controlling other insects. Oftentimes, some insecticides will have a broad efficacy across a number of different species. And so even though we may be treating for a different insect, the constant exposure of the armyworm to different insecticidal mode of actions, to a single insecticide mode of action, can eventually select for a resistant population. And that's where including a commercial BT in your spray program can help to control those insects, because there is no known cross resistance between a commercial BT and any other insecticide class, which is really fascinating and really unique within insecticides. And this is borne out over 50 years of different insecticidal mode of actions being introduced into the market. And so finally, excuse me. And so finally, the commercial BTs work exclusively against the larval stage of the caterpillar. The larval stage of the armyworm. They'll have no effect on eggs or on adults. And that's really full stop. The mode of action is through ingestion. And so it is only the larval stage that can really be intoxicated by the commercial BTs. And so that's why as we'll discuss at the end of the presentation, timing and coverage are just so important when it comes to commercial BTs. And so we covered a lot of ground there. So if anyone has any questions.
Yes, Daniel, there's lots of questions. There's already 20 answered.
Okay, great.
So Chris is really probably getting a sore wrist at the moment, I think, a hand there, like typing out answers. But a couple questions here. Are BTs temperature dependent, and what is the temperature range to have the maximum efficiency or efficacy?
So interestingly enough, the BTs are not temperature dependent. The caterpillars are temperature dependent. The caterpillars won't eat foliage when it's too hot or too cold, and so for that reason, the optimal temperature is when the insect is feeding.
Okay.
Excuse me. The commercial BTs aren't affected by high or low temperatures necessarily, like from the environment. So it's really has more to do with the insect than it does the products.
Okay, there's another question here around the shelf life. Now you might get, if you're going to cover that later, we can leave it. But there was a question just around what is the shelf life of a BT pesticide, biopesticide?
Yeah, so that'll be very specific to the product. I think it's probably best that you refer to maybe the sales rep or the retail who's selling the products to ask them what the specific shelf life is. Okay. With respect to the products that Valent Biosciences sells, we have a shelf life of at least 2 years.
Okay.
I was fortunate enough to collect some of this dipel DF from our customers in Japan, who oftentimes will think in very long term. This product was over 8.5 years old, stored in a temperature controlled office, and after 8.5 years, still was as potent as the day we sold it.
Okay.
So stored properly, they're incredibly stable.
Okay, stored properly. And so I've got a question here. Yeah, which is always probably the case with lots of things. Daniel and maybe Roma afterwards, but there's a question here from Andy, and thanks for joining us, Andy. It's despite the advantages of BT over chemical synthetic insecticides, one of the concerns is the consistency in their performance under field conditions, which are related to the mode of entry through ingestion and the behavior of the larvae, such as fall armyworm, where big larvae tend to live inside the whorl and are protected. Are there any special efforts to cope or address these problems?
So I think the most important efforts that can be focused on how to get better consistency is twofold. And I'll mention this at the end, but the most important aspects are timing and coverage. A commercial BT will only be effective on the leaf for somewhere between 3 to 7 days, which means that you have to time that application perfectly. And that's where having an organization like this consortium put together by Grow Asia is so critically important to come up with tools that can help us understand the life cycle better and time those applications better. best. I think that's really the number one source of variability in the field, is that timing and coverage more than anything.
Excellent. Roma, have you got anything to add to that?
No, I don't think I can. Yeah, Dan's right. I think one of the things we should always acknowledge for biocontrol agents is they aren't highly toxic and they aren't highly persistent. So you've got to be really good about when you apply them, really understanding and monitoring your pest population well, so you get the timing of the application of your biocontrol agent with the timing of the pest. So you have to really be good at understanding that biology. Plus, you have to actually have really good application equipment. So really think about what you're doing, about your spray volumes, about you've got good quality tanks and good quality applications. And I think actually this is something that Daniel's probably going to touch on in the second part of his, in about a month's time, isn't it? And how to actually get the best out of these products in the field and how to use them well.
Yep, that's right. And we're hoping to bring some video footage as well of actually in the field and how to do that, everyone. So hopefully you'll join us in a month's time. Daniel, if you could continue on, because I know we've still got lots to pack in.
Sure. So I'd like to change directions here and start talking about the importance of quality in commercial biopesticides. Now, when you think about quality of a product, I feel like it means something a little bit different in commercial BTs and just biopesticides in general. And that's because we're growing a microorganism, which requires very special conditions to get really the high potency that we need to be effective in the field. And so for that reason, the source of that material is critically important. Who grows it, how they grow it, what are the quality controls, all contribute to actually how effective the product is. And so as an example, I was gonna have a demonstration. I don't know if we have time for it, but there are available on the market cry protein test strips. So these are kind of like, they look very similar to maybe a COVID test that you might have taken, where you have on the top here a positive control band that just says that the thing is working. And the second band will light up in the presence of a cry protein. And this is really critically important throughout the region, because oftentimes low quality commercial BTs will have very little cry protein. And as we've mentioned a 1,000 times already, the cry protein is by far the most important aspect of the mode of action. And a lack of cry protein means a lack of toxicity. So we have this tool, this is these cry protein test strips, which can very easily be used to determine whether a product contains cry proteins or not. And so I actually have here, I'll just pause this in a second, but I have here an example of what can be done. And I've sort of done this on the back of a truck before as well. So it's a neat tool that you can really pull out into, you can take out into the environment. How do you stop sharing? Am I sharing the screen right now?
Yep, you are.
I am, okay. How do you stop sharing? Oh, there we go.
There we go.
How about now?
I think it might take a little bit of time. Yep. Is that working now, Daniel? I can see it.
Okay, there we go.
Yeah.
So what I have here are these cry protein test strips. So we get these from, in this case, Romer Labs, but there are a number of different laboratories. These are often used to identify the presence of cry proteins in seeds for those who are interested in growing non-genetically modified crops. Now, I just want to be very explicit. There are no genetically modified commercial BTs available anywhere in the world. It just so happens that the cry proteins in commercial strains are so effective that it became really a primary insecticidal component in BT crops. They really took it straight from the insect. And so what I have here is, this is just some water and this is a, what do you call these? A microcentrifuge tube. So 1.5 mil tube with a cap. Now, it just so happens this cap holds about 100 milligrams of a commercial BT, which is very convenient. So these kits will come with a buffer, an alkaline buffer, and that's really important because the if you remember correctly, if you remember from before, the caterpillar gut is specifically alkaline. And in fact, those cryoproteins won't be dissolved. Those crystals will not be dissolved unless they are dissolved within an alkaline environment. So we can just toss that in there, give it a shake. And when you're doing this, it's actually an interesting, it's interesting to watch because this is, I have here, this is some of a bag of a dipel from the US market. And it's kind of hard to see, but there are, you can see kind of the product floating in there. And that's because commercial BTs are not soluble. You won't see them completely dissolve into the material, just completely dissolve into water, because again, the crystals and spores are insoluble. They're actually quite large. If a human hair is about 100 microns wide, then a BT crystal is about 1 micron wide. So about 100 of them side by side will give you one human hair. So I have here, took some of this material. I'll pop in a cryoprotein test strip, and then within a few minutes, you have your result as to whether it contains a commercial BT or not. And to save time, I actually had run one of these earlier this morning. And as you can see here, the top band is the positive control. So if it doesn't light up, it's not working. And the bottom band shows a BT, shows that this contains a Cry1A type protein. Available on the market today are Cry1 and Cry2 type protein test strips. And what we found with using these test strips is that there are, unfortunately, fake commercial BTs on the market, either those that are adulterated or those that are of such poor quality that they in fact are unlikely to control any caterpillar pests at all. And so having a tool like this, can be extremely valuable for identifying those products in the market, ensuring that you're getting a good quality product. But it also highlights the importance that unless this product contains the cryoproteins, a commercial BT, it will be ineffective. And so there are a number of other watchouts to keep in mind when looking for a commercial BT on the market. The first is solubility. As I mentioned before, BTs will not dissolve. In fact, if you can still see in this image, there might be a little bit of material that's floated to the bottom. And that's actually expected because commercial BTs are insoluble. If a product dissolves completely, or if the product is clear to start with, it is not a BT. And that's just full stop, it's not a BT. Secondly, the speed of action. Insect mortality from a BT can take about 24 to 48 hours, although larvae will stop feeding immediately. Now, a BT claiming quick kill is either adulterated with an insecticide, or actually some strains are known to contain what's called the beta exotoxin. It's a Bacillus toxin that is in fact not allowed in most countries, as it's acutely toxic to people and can actually cause skin rashes. We're required in most countries to prove that our commercial strains do not produce this. Non-target toxicity is also an important watch out. BTA and BTK will only affect caterpillar larvae. They will not impact non-target organisms. So if a BT label claims activity against other arthropods, like ants or termites, it is not a BT, or it's adulterated with another insecticide, which hasn't been listed on the label. So these are just a few things to watch out for as I hope that as biocontrol becomes more mainstream, the kind of oversight and regulation that ensures that everyone gets the best kind of products they can, gets better over time. But in the meantime, we really have to be vigilant and understand what makes these products work and what makes them, what makes a good quality BT. And so I'd just like to end with the fact that growing your own BT, for example, is I think something that a lot of folks have thought about. And unless you can get the conditions just right, there usually isn't enough cry protein in just backyard BT to make these products effective in the field at the kind of concentrations you're going to want to use them. And so for that reason, having a really reliable source of commercial BTs is critically important. Now, one misconception that I'd really like to focus on is that spores are not the primary mode of action of the commercial BT, but you may actually find it on the label, depending on where you live. For example, on the left-hand side, this is a Dipel SC from Indonesia, where you can see that the active ingredient is listed as the BT Kerstaki strain with 8 times 10 to the 7th CFUs, which is a reference to the number of spores. There's no other reference to what the active ingredient is. On the right-hand side is a Zentari label from Thailand, where the product is labeled in something called DBMU, but also in CFUs. And this is a reference again to spores. Now, when I wanted to bring this up because oftentimes when we're choosing a pesticide to use in the field, we'll oftentimes look at how concentrated it is, how much of the active ingredient is there, and then we can do the math in our head and find out what the best price is. But in the case of commercial BTs, it's really not the information that's on the commercial labels the information on the active ingredient in the product is actually a bit deceptive. And that's because of the complexity of the organism. When commercial BTs were first introduced, regulators didn't know what to do with them. It's not a traditional active ingredient. There's a lot of different components. And so across the world, different regulatory agencies sort of picked at and decided on different things to look at. In some cases, the result was spore count, because it's something that's easy for regulators to confirm. However, comparing spore counts on the label does not mean that a product with more spores is more effective. And that's because it's the crystal proteins. And in nowhere in the world are the cry proteins themselves actually listed on the label. So again, just to be very clear, comparing products based on spore counts is not a really good way of comparing products. You'd want to look at the labeled application rates more than anything. And just as an example, this was an interesting study we just recently done at Biopesticides, where we essentially took the Dipel DF in that microcentrifuge tube, blended it up, and then spun it down. So we had the solids, which were collected as the pellet, and then the supernatant, which is the liquid above. And so starting on the left-hand side, this is a leaf dip assay using second instar cabbage loopers. And you can see in the negative control by 3 days after infestation, there's not much left of that leaf. The positive control here with 1 gram per liter of Dipel DF effectively controls those insects by 3 days. And in this treatment, we have both the spores and the crystals because it's the fully formulated product. Now in the supernatant, so this is the liquid above, we still have some spores that won't spin down. And interestingly enough, you can see that they do have effects on the insects' feeding habits, but they don't kill them at all. The graph above is a % efficacy. And you can see that no insects were killed, yet it did seem to have some mild effect on how well they ate. When we then filtered that supernatant, removing any spores that might still be there, you could see we returned to that basically the same effect as a negative control. And finally, the pellet, so all the solids, the crystals, the spores, any of the soluble proteins, that's where all of the activity is. And so even though spores may be on the label, they are not a critical part of the mode of action. I'm sorry, they are not alone insecticidal, and therefore not a good way of comparing BT products. So in summary, what we've talked about today is all the different aspects and the complexity of these commercial BTs, with the crystal proteins, the viable spores, and all the other soluble synergists that all work together. And so the method that was determined to be the best tool for assessing the quality of a commercial BT was something called the international units, which is derived from a live insect bioassay. And so this video here actually shows the the cabbage looper bioassay that we conduct at Valent Biosciences. So we maintain a colony of cabbage loopers, actually the same colony for probably nearly 50 years. Here you can see the eggs are collected. They'll then be hatched onto a media, and then they'll be infested into media that's dosed with the products. And in this way, we can look for the LC50, that's the lethal concentration that will kill half the larvae in the treatment. And that will tell us the quality of the products. And this results in a metric known as international units. So it's important to note that it's not very common to test against a live insect bioassay. This is quite unique for Valent Biosciences. However, in most cases around the world, a quality control metric has to be correlated back to this type of study. And this is really important because this is really the only way where you can account for all the different metabolites working together is against that live insect itself. And so international units, or IUs, are truly a unique way of measuring BTs, but most importantly are not related to field efficacy. I wish I could tell you that there's a lot of important information here on the front of the label under active ingredient, but it is for the most part very deceptive. Those international units are only based on a laboratory bioassay against an inbred population of cabbage loopers, only cabbage loopers. It does not reflect efficacy in the field, as this is an inbred population. And it also does not reflect efficacy against any other species, as the only thing that can be called international units are against cabbage loopers. And finally, this metric then is really only used for quality control to test the complex mix of crystals, spores, and synergists. And so again, when comparing commercial BT products, you might notice some on the market which have potencies of 16,000 international unit. Another product that may have a potency of 30,000 international units. And in fact, those products may be just as effective in the field. What will matter more in the case of fall armyworm is the specific blend of cryproteins, something that just isn't on the label, which is really unfortunate. The information that's on the label here about the active ingredient just doesn't give you a really good idea about what's in the bag or what's in the bottle. And so using spore counts or using this biopotency, these international units, to compare BT products and make a choice about which one you want to use is actually not the way to go. What you really want to think about is the reputation of those commercial products and the reliability that you see yourself. So when choosing a label that you may not recognize, something new on the market, it's important to look into the reputation of the company manufacturing those products because the quality controls are so important to make sure that you have that high concentration of cryproteins. So again, it's unfortunate, but there's really not a lot of information on the label except application rate that can be used to compare across application rate and subspecies, of course. And so I could pause there for questions again.
Great, Daniel, and lots of questions still coming in. So yeah, people obviously got a lot to ask here. A couple of questions, and maybe I can join one or two into one question. But it was really about the access to these test strips, the cost of them, and also would it be smallholder farmers doing this, or do you think it's more realistic that officials or extension agents or regulators would be doing that type of thing? I mean, how realistic is it for smallholder farmers to be doing that?
I don't know if it's so realistic. I think I wanted to just show this.
No, it's very good, but I was just wondering, I think people are very interested in it. I mean, I think you would see maybe extension agents having access to that and understanding what they're doing, I guess. And how much is the cost of that?
So we buy them in these special this special small pack that they made for us, and I think it's about $2 or $3 per strip. They're not cheap for sure. Yeah. But they're an important tool.
Okay, excellent. Now we're just going to go down into maybe a few more technical questions here around firstly, I'll ask this question. Any results of BT for controlling rice leaf fodders or stem borers? Is BT effective?
I think Chris might have direct experience with this one. We do have efficacy against the leaf folder, but I believe the stem borer is where we get into, we have that issue of the insect boring too quickly and not eating the outside of the leaf.
Excellent.
So that's an example of where the habit of the insect is really important, because again, BTs are not systemic and are not, they can't move into the plant.
Okay, so that leads into another question that's quite common, is like, for the fall armyworm, when are you wanting to, when's the best timing to sort of apply this? If it's the caterpillar, what stage and when?
Absolutely. The best timing for fall armyworm is as close to hatch as possible.
Okay.
The first and second instars are the most susceptible. So when the caterpillar is so small, it's hard to see. And so that's where scouting and having models that are reliable can be so important. So when pheromone traps are available, if they are available, using them to keep track of whether the moths are flying into your fields can be really valuable. Because just understanding when that brief period in the first and second instar, that's when you want to target the application.
Okay, great. And I know we'll talk about that a little bit further and as a bit of a taste for the session in June. So we'll come back to that, probably the specific application and ideas around that. But here's another question. Is there any relation between water quality and the BT efficacy?
So that's something we'll cover in the next presentation. There's a little bit in this one, but the water quality actually apparently has a very little effect on the commercial BTs.
Okay.
There are examples where the cry proteins are alkaline soluble. And so there are regions I know where where BTs are applied for the bunch moth. And in those cases, the water can be extremely alkaline. Now, the VBC commercial formulations actually do a very good job of buffering the water. However, checking the tank pH after introducing the BT is very important, because if the water does remain alkaline, it'll actually dissolve those crystals and make them ineffective in the field. So that's really the most important aspect.
Okay, and we'll talk about that further, but that's a good good taste of what's to come as well. Another question here, a more generic question around, are BT proteins allowed in organic agriculture globally?
Absolutely. And this is that misconception of the overlap of GMOs and commercial BTs. The commercial BTs are the natural source of cry proteins.
Yeah.
Most of the VBC formulations are in fact organic certified, including the one that I tested today.
Okay. Great. Now I'm going to let you carry on, because we're tight, a little bit tight on time. Not really. We're actually perfectly timed, actually, with 5:10. So, but I want to leave a bit of time at the end, Daniel, because I know that there's quite a few questions still coming in. And what I'm quite keen is you're going to give a bit of a taste, a little bit of the information which is going to come up in the next session. So this will also probably raise a few questions. So if you carry on, and then we'll have a bit of time at the end for finishing all the questions that come up. And thanks for all the great comments, everyone that's coming in as well.
Absolutely. Okay, so what I'd like to end with are what are the two most important aspects for getting good field efficacy with a commercial BT, and that's timing and coverage. BT retain their peak activity for only 3 to 7 days. Excuse me, so timing is critical. Targeting early instars is extremely important, especially with borers before they become inaccessible to the product, like the rice stem borer we talked about. If not already a common practice, regular scouting can not only be beneficial for all aspects of crop protection, but especially for getting that application timing just right. And that's where, as I mentioned before, having access to predictive models whenever possible to time that application is really extremely useful. We've as VBC, we've helped to develop some of these models. For example, for the sugarcane borer in Brazil, and being able to map the moth flights in your own field can allow you to time that application so precisely in the case of that particular insect that even a commercial BT, which, as Roman mentioned, these products are usually less toxic than these the traditional chemistries, but getting the timing just right means that even the slightly less toxic pesticide can be just as effective. And this is a study from so Mato Grosso state in Brazil. Along the left-hand side of the table, you see these are 1st through 5th instars, and across the top are different caterpillar species, in this case collected from the field, with a application rate of a product, I believe this one was Zentari, at either 400 or 600 grams per hectare. The boxes that are in green indicate 95 to 100% control. So that means all of the insects of that instar were killed. And anything in red means less than 70% mortality. And so as you can see very clearly, 1st and 2nd instars across the board are the most acutely susceptible to commercial BTs. After that, it becomes very specific to the insect. For example, on the far left-hand side, the Chrysodexis includens, which is the soybean looper, is actually can be affected by commercial BTs up until the 5th instar later in their life. While in the case of the fall armyworm on the right-hand side, in this case, a population from Brazil. So of course it can vary from location, even vary from field, to be honest. It was up until the 3rd instar where the commercial BT could effectively control that insect. So this just goes to show how important it is to time that early application. And also, when you are scouting in the field and you find mixed populations, keeping that in mind that the commercial BT will control the early instars, but not necessarily the later instars, may inform whether you use different rotation partners or different tank mix partners. And secondly, the second most important aspect of field efficacy is coverage. And this is because BTs are not systemic. Coverage is critically important to protecting foliage and maximizing the value of every application. Only foliage that is directly sprayed is protected, full stop. For best results, it's best to ensure full coverage with the lowest reasonable volume. Oftentimes we use this kind of catchphrase of spraying until the foliage is dripping, but not excessive runoff. If the water doesn't stay on the plant, neither does the product. And higher volumes may be necessary under heavy pest pressure to ensure complete coverage. This is where it may be important to use higher volumes if the insect is moving into the leaf whorl to ensure that you get enough accumulation of the commercial BTs in that leaf whorl. And this is just a taste of what we'll discuss in the next session, which will be coming up in June. In that session, we'll talk about really where the rubber hits the road, when to apply based on scouting, so using scouting to make decisions, storage, handling, and PPE, application equipment, rates, spray volumes, tank mix compatibility, how to apply the products, and then how to verify efficacy. These are all things that we'll cover in the next session, and so I really hope you'll join us for that session as well. And I'd like to end with what I think are, maybe the most useful top 10 considerations for using BT in the field. I won't necessarily read through all of these, but it kind of highlights some of the things I've talked about and also some things that we'll talk about next time. For example, hit them early, making applications against early instars. Watching the tank pH, number 5, is important because if the tank is alkaline, it'll ruin the product. Tank mixing can normally be okay with insecticides, fungicides, and other additives. And number 9, I think is one of the most important misnomers, is that BT, they'll actually last for a few days. So they can be applied day or night. I know some folks will only apply a BT at night thinking that it makes a really big difference, but in reality, actually doesn't make that much of a difference. So I'll leave this up for a minute in case you want to copy it, but that ends my presentation. And so I'd like to take any more questions, and I truly appreciate all of your attention.
Yeah, Daniel, that is fantastic, and at perfect timing, which I knew you would do. It's always difficult, everyone, when you're a moderator, to always get the timing right. But it's fantastic when you have a speaker that has definitely prepared very well. And I really enjoyed your live demonstration there, by the way. Very good multitasking. So congratulations on that. Quite a few questions here, and you've got lots of good feedback, Daniel, that's been coming through in the chat too, just to tell you that. this question's come up, and I know you answered this, but it's come up a couple of times and still coming up. So I thought it would be good just to go back on it. And it's really around why you have to get them in those early instars. So why is there less mortality in the 4th and 5th instar? And I know you addressed this, but it would be good just because I can see it's coming up a few times. Let's just go over it and just make sure everyone's really clear around what's happening here.
No, it makes sense. And actually it's the same reason why, I think if you read the labels of most insecticides, for the most part, the suggestion is always to target early instars. And that's just because of concentration effect. A small larvae really is just much smaller and has to consume less material to get a lethal dose. A 4th or 5th instar larvae that eats just a normal application rate of commercial BTs, while it may cause damage, make them sick, make them lethargic, actually, interestingly enough, offset them in the reproductive cycle. So they won't necessarily be able to mate with the next generation, but they'll still survive because they can just outgrow and survive the effects on their stomach. Yep. And again, that goes for really all insecticides. The bigger the insect, the more it takes to kill them. And in the case of BTs, you can poke a few holes in a small larvae and have a significant effect, but that same number of holes in a much larger larvae will make them sick, but won't kill them necessarily.
Yeah. Excellent. And so here's another question that comes through a lot too. So, and thanks for that answer. Makes sense and actually very interesting as well. Different generations as well and the impact there. Here's a question here, and there's 2 parts to this. Firstly, can BT products be mixed with chemical insecticides to kill larvae, eggs, and adults all together? And then the second one, could it be mixed with fungicides? And are there any exceptions?
Yeah, so there's really 2 considerations, and that's the effect that these products will have on the proteins themselves. So strong oxidizers should not be mixed with commercial BTs because they can degrade the proteins. There's some fungicides that are just very strong oxidizing agents. That being said, most insecticides are not. And so actually commercial BTs appear to be, of course, I can only speak for the ones that we've tested, but we've tested quite a few at VBC over the years. And there's no incompatibilities there. In fact, it's really beneficial to mix commercial BTs with other insecticides because again, it probably is likely there will be mixed populations in the field. So getting effective control may require an additional step, having something else. So generally speaking, there's tank mix compatibility across the board. That goes for the chemical compatibility. Physical compatibility of the formulations should always be tested separately in a jar test. So mixing the amounts in a jar and then looking for just physical incompatibilities is important.
Yeah. Okay, good. And here's a question here as well around, once you've sprayed the BT products on the plants, will the BT start to propagate? Does it have any antagonism on the surface microbial composition of the leaf surface, which could lead to other problems?
Now, interestingly enough, commercial BTs for the most part are so specific to this ecological niche within the insect that they almost never grow in the environment. I met a researcher recently who had applied commercial BTs to a field. It was once 20 years prior, and every few years he would go out in the field and test the population of the commercial BT that he could find in the soil. And what he found is that after something like 20 years, at most, there had maybe been a few generations of outgrowth of the commercial BTs. So they really don't grow very well in the environment. Their preferred environment is inside the caterpillar. So they're not going to grow on produce. They're not going to grow in the bottle. So if you see a bottle inflate or something like that, it's not because the BT is growing, it's because there's something else in the bottle. And they're not really going to grow in the soil. They won't necessarily accumulate in the soil or anything like that. They're so hyper specific to caterpillars that it's really the only place they efficiently grow.
Yeah. Okay, excellent. Very interesting. And here's one, I've got a question coming in here around, you say the IU cannot be used to determine the bioefficacy of BT products, but how should we do that then?
Yeah, it's not a I'm very sorry, it's not a very satisfying answer, but reputation and experience is really the best way. We do have tests for quantifying the amount of cry protein in a product, but it's not very common.
Yeah.
So really it's knowing where you're getting these BTs from,
Yeah.
having a label that you can trust. It's unfortunate this is how it is, but this is just how the industry is regulated. Yeah.
Okay, Daniel, I'm gonna ask you a question. I'm gonna ask Roma probably the same question, or to add to it, for sort of the last kind of question. And I guess this is a question around, we've got a lot of smallholder farmers in this region making up the bulk probably of the farming community. I mean, how cost effective is this as a form of control for fall armyworm, and how accessible is it to smallholder farmers in this region at the moment? Not to say that there may be opportunities to make it more accessible, and I think that's what we're trying to do through the Biocontrol program, but what's your thoughts on that for now?
Roma, did you want to comment?
Oh, sorry, I thought it was to you, Daniel.
Oh.
Oh,
I don't know.
Let's let Roma go first. And then she's probably.
Sorry, I was waiting for you to. Yeah. So it's a difficult question, because I think, as Daniel has pointed out, what's really important with microbials, and to be honest, any plant protection product, the quality of that plant protection product is important. And there used to be this concept that biocontrol agents can be produced cheaply and easily, and everybody can do it on their farm. And the truth is, you can't. They're much more complex than that. It's much harder to maintain the quality. It's hard to maintain that you've got no contaminants in there. So I think we need to sort of move away from that concept of them being microorganisms being a sort of cheap solution for crop protection. But what they are is, when you've got a good quality product, is that they are effective and used well. So I think some of the ways in which smallholders can save money is to get good information about how to use the products, so that when they use them, they're using just what they need, is applied at the right time, and getting the most out of it. Yeah. I think mechanisms to make them available is something that governments need to think about in their policies if they have a particular focus on smallholder farmers.
Yeah, excellent answer. Thanks, Roma. Did you want to add anything to that, Daniel, or did you want to add anything else that comes to mind for your last statement?
Sure. I think the only thing I would add is that with respect to this particular question, is that this is where tank mixing can make the application of a biopesticide more cost effective, and oftentimes how commercial BTs really get out in the field when cost is an important issue. But what I'd like to close with is the fact that it is important to find ways to introduce, if not commercial BTs, other biopesticides into the control of fall armyworm in general. Yeah. Resistance is a huge issue. It's going to be a huge issue. We know this in the Americas, and I believe even some of the resistance that was already inherent within the insect, it carried along with it into Asia and into Australia. And so having different tools and using those different tools together is really at the heart of having a good and effective control program. And I think BTs are an especially good tool just because of how unique they are, and because of the other value that they really bring to the field.
Yeah, that's a brilliant summary. Yep.
Yeah, just to add to that, I mean, this is the aim of the WHO fall armyworm project, that the way to manage this pest is an integrated pest management approach. There isn't a single solution. It's about layering all the solutions together. You've got really good monitoring, you really understand the biology of the pest, and then you look at all the tools that you've got available, and you use those tools well, but using them at the right time, but just what you need at that right time. So yeah, Daniel, this is about integrated pest management. I think the days of sort of you could spray something and walk away and forget it are long gone now. We've got to have a much more integrated approach to crop protection.
Excellent. Thanks, Roma. And thanks, Daniel. Brilliant presentation. Very comprehensive. And I'm really looking forward to the next presentation coming up. Based on this one, it's going to be fantastic. Really like your enthusiasm as well, I have to say. It's quite nice to see the beautiful crystals. You almost have quite a passion there explaining some of the more technical stuff of the product and how it's used. So thank you very much. I'm just going to end your bit here, and what I'm going to do is just ask Fang Zhong from CABI just to provide some closing thoughts and a summary. And while you're doing that, I'm going to share my screen and just get back to the presentation. Fang, if you could unmute yourself, and I'll hand it over to you.
Oh, thank you, Alison. Congratulations, Daniel, for your excellent presentation. I'm very impressed with the information you provided. Of course, this also reminded me, 10 years ago, I was working on BT in DPR Korea. We tried to produce BT with the semi-solid fermentation method, as you mentioned. Quality is really an issue. Contamination, you can imagine with the semi-solid fermentation, it's much more difficult to control the quality. But even with that, I was still, remembered how successful, with the products to spray by farmers in the field to control diamond back moths at that time. Now we are going to work on the fall armyworm in the region, particularly. I see. I don't want to repeat that you have already mentioned the timing, this quality, how important, and also how to select the right species strain, all these factors. You have given a very good summary. I like it very much. Actually, I'm eager to see the next presentation next time. Alison organized in end of June. That's a little bit long, I would say. We should shorten this if it's available. So to use the momentum. So and also I see how enthusiastic from the participants to look forward to see this to use the BT or to apply this in the field. I see the questions, and the very typical questions comes from the region. I do thank the group from Daniel, from Element, and also Roma to give very crisp answers to each questions. Thank you all for this excellent work. From KB side, I would also like really to add our thanks also to Alison and Grow Asia to organize such a wonderful event for us. With that, I would like to, to finish. Hand over back to Alison, please.
Excellent. Thanks, Fang. And thank you for the summary of your key messages and the enthusiasm. We do need to leave a little bit of a gap to the next webinar because we really want to get into the field and actually get some video. So that's why there's a bit of time. So sorry for the wait, but people can anticipate that it will be a very good session. Big thank you to Daniel, Chris, Valent Biosciences, Sumitomo for your preparation support of the webinar. Also, thank you very much, Roma, for joining us as well. It's very good to have you and to have your input. Looking forward to seeing you on the June 30th. So thank you very much for joining us today, and thank you for all your questions. We have a lot of questions there. We will post a copy of the PDF and a copy of this video online within the next week. Just remember, you have other sessions, so please sign up. You can see that on our events page. We do have a Biocontrol regulatory masterclass, particularly if you're working in government around regulation. We would love to see you there. So you can contact me if you'd like more information on that as well. So that really draws us to a close today. Thank you so much. And thank you, Cabby, for doing this together with us again. We really appreciate your support. So take care, everyone. Have a safe couple of months, and we'll see you back in June. Thank you. stop the recording.